Welcome back. In the previous lecture, I introduced DNA as a genetic material and as something that gets expressed and duplicated in precise ways. And I also discussed how spontaneous and externally induced DNA changes underlie the development of cancer. In this lecture, I want to describe things that actually do the DNA damage, namely carcinogens, or things that cause cancer. I will also describe some carcinogens that don't damage DNA and yet cause cancer, and finally discuss some DNA changes that are not inherited, called epigenetic changes, that are involved in the expression of genes and also underlie cancer. The multi-step model that I described in the last lecture involves sequential mutations happening to a cell that, in the end, once mutations have piled up, is a cancer cell and a cell that can spread throughout the body. Just to review, initially chemicals that cause damage or radiation that cause damage go into a cell and cause genetic changes, and we have what is called an initiated cell. It's got a couple of genetic changes. And then more changes happen to get a lesion that's pre-neoplastic, and then a tumor that's malignant, and finally a tumor that can spread in the body. To review, normal sequence of DNA might have a C at this location. Remember, DNA has two strands, A, C, G, and T, and it might have a GC at this location. And in the mutated DNA, there's an AT at the same mutation, and that's the mutation. Mutations happen all the time. And fortunate for us, mutations can be repaired. There's a repair mechanism that scans along DNA for mispaired or altered bases. For instance, if there's an AT base pair normally at a location, and all of a sudden the T changes to a C, and now you've got an AC, an A in one strand and a C in the other strand, and they're not quite matching, the repair system goes along and says, oh, problem, and it takes the AC out and puts in the proper bases. So this is the graphic of DNA repair, and most spontaneous mutations are repaired. The mutation is removed, and then the proper bases are put in. To give you an idea of the magnitude of changes that can happen to DNA just because it's DNA, just because it's chemical, take a look at these numbers. 3 billion bases in the human genome, and every day about 16,000 bases get mutated. You can imagine it's going to pile up over a lifetime of many days, and we're going to be loaded with mutations. Fortunately, 80% of these DNA changes are repaired by this repair mechanism, and there are several repair mechanisms for different types of DNA damage. Now, there are two aspects of cancer that involve DNA repair. In some cancers, the repair system itself isn't working. It's mutated. The repair system is genetically determined, like many other things in the cell. It's one of those biochemical things. And so mutations can happen. It's DNA. And then if mutations happen there, then other mutations in DNA are going to be piled up because there's just going to be an accumulation because there won't be repair. And this indeed happens in some key genes that regulate cell division. And if that happens, then there'll be more cell division and you could have cancer. You'll see later on in the course that this indeed happens in colorectal cancer, for example. In other cancers, the mutation rate is so high it overwhelms the repair system. There's just so much damage, the repair system can't keep up. This is going to happen if there is a chemical or radiation mutation that's very strong and it's changing a lot of the DNA very rapidly, and the cells are dividing very rapidly, so the mutation kind of gets permanentized and made permanent in the DNA. The carcinogens, the things that cause cancer that mutate DNA, are of two types. First, chemical carcinogens, and second, radiation. So I'm going to talk about both of these in turn. First, chemicals. We are exposed every day to tens of thousands of chemicals, natural and synthetic, that come from outside of the body. You know, one of them is H2O, but there are lots of others. Caffeine, if you drink coffee, coffee has hundreds of molecules in itself. A chemical that reaches a cell in the body after being exposed on the outside is not usually the same as the one in the environment. That chemical has been transformed. And the transformation happens in an organ called the liver. Let's study this transformation for a moment. Here's a location of the liver in case you didn't know. A molecule enters the liver typically from the digestive system, or it can be from the blood system, either way. but it ends up in the blood and goes into the liver where it gets transformed. Now, you don't need to be a chemist here. I'm not asking you to be a chemist. I want you to focus on the transformation of this molecule. Here's the molecule itself, and here's the transformation. I'm circling it. It's an OH group, OH. And when a chemist sees OH, the first thing they say is, oh, well, not really. They say, this molecule has a different property because it has this OH. And the different property the molecule has is it's more soluble in water. It can dissolve in water. This system of transforming molecules in the liver to make them more soluble has evolved over over eons of biological time because we don't want molecules to accumulate in the body. We want to make them water soluble so we can excrete them. Every day we're exposed to thousands of molecules, and if they didn't dissolve in water, they might pile up in our tissues to toxic doses. Instead, this liver system changes the chemicals to make them more soluble so they can be excreted in the kidney. So the pathway is food or into the intestine, which then goes into the blood system, which then goes to the liver, gets changed back to the blood, into the kidney, and excreted. Good idea. However, the molecule, when it's in its changed forms, it has these extra oxygens in it, is also more reactive. It can do things. It can bind to DNA or change DNA. So the race is on, right? The molecule is on its way out to get excreted in the kidney, but as it goes out, it may get into some cells and cause damage to DNA. I'm going to describe two carcinogens that act on DNA, and they're transformed. One of them is transformed by this system, the other is not. One binds to DNA, and the other actually changes DNA. The first one I'm going to describe is called aflatoxin. Aflatoxin is made by a fungus that coats seeds such as corn and peanuts, stored seeds, dried seeds. Aflatoxin is a major problem in the food and agricultural industry. The levels of aflatoxin that are tolerable are extremely low, and they're regulated by the government. The governments always are inspecting seeds for aflatoxin levels. Aflatoxin gets changed in the liver system. Now look at the graphic, and you'll see the change that happens. Here's the aflatoxin molecule. And here it is getting changed. It's not an OH, but an O. See that O that's there? And this makes aflatoxin more soluble in water. That's great. It's going to get excreted in the kidney. Unfortunately, it also makes it react with a G in DNA. So here's the aflatoxin bonding to G in DNA. Here's aflatoxin, and here it is attaching to G in DNA. It essentially covers the G in DNA. The G in DNA now has been changed irreversibly, and so what happens? During DNA replication, when DNA becomes duplicated, the duplicating mechanism comes along and it sees this coded G and it says, chemically speaking, what is this stuff? It's not a G because it's got this huge aflatoxin thing is, I don't know what to do, but I got to keep going because remember, DNA molecules are 100 million nucleotides long, got a lot of work to do to replicate the whole molecule. And so it keeps going and it just says, oh, I'll just put anything in. Instead of putting a C, which you would put opposite G, it's going to put in something else. So in this case, for example, here's the G. The G has been coated with aflatoxin, and instead of putting in a C, the replicating machinery puts in an A, and now you've got a mutation, right? And in a second generation, a second DNA replication, this GC pair up here becomes a TA pair. That's the consequences of aflatoxin mutation. And this can be repaired, but the repair system sometimes is overwhelmed, as I said, or the repair system isn't working. The second molecule I want to describe that's a mutagen is nitrous acid. Nitrous acid is a common chemical group in natural and synthetic molecules. It's one of the molecules that's formed. Remember in the first lectures I described salts preserving meats and fish, and there's a reaction that happens that releases a carcinogen in the stomach. This is it. And this is what happens when nitrous acid reacts with DNA. Again, I'm going to show you the chemistry just to show you what happens. Here is a molecule of adenine, A, one of the 4 bases of DNA. And the A has on it an amino group, an H2 group, and that's changed into an oxygen group by nitrous acid. And when this change happens, it's an irreversible change in DNA, and the base pairing characteristics change. Instead of this A bonding to a T, instead it bonds to a C, and you're going to get a change in DNA of a base pair change. So here's normal DNA with an AT base pair. And now the A is changed to an X, hypoxanthine, the name of it is. The X doesn't normally bond with T. Instead, the replicating machinery comes in and puts a C in. And so, of course, one more round of replication, and you made a GC pair where once there was an AT pair. So here in detail is the chemistry of two substances that are very common in causing mutations, and these mutations can end up in cancer. Many chemicals have been examined for causing cancer and DNA damage. They're natural chemicals, and they're synthetic chemicals as well. It's very hard to imagine that we can look at every molecule we are ever exposed to, but people try. There's a central database at a place called the International Agency for Research on Cancer, based in Lyon, France. And it collates data from all over the world, from epidemiology, from DNA experiments, etc., and classifies substances in terms of mixtures like tobacco smoke or chemicals or radiation to say what causes cancer. And they classify these things into 4 groups. Group 1, carcinogenic, definitely to humans. I think aflatoxin is going to be on that list. Group 2A, probably carcinogenic. Evidence is pretty good. Group 2B, possibly carcinogenic. Group 3, we don't know, not enough evidence. Group A has, Group 1 has 79 chemicals on it. Here are some of the chemicals in this graphic of the 79. First in the list, of course, is aflatoxin as you look down the list. Some of these are natural chemicals, such as, of course, aflatoxin, estrogen, right? That's a natural compound. There's some others. Others are elements in nature. For example, chromium. That's famous from a movie called Erin Brockovich and the book based on it. Others are drugs, like cisplatin is used to fight cancer. Dioxin, you may have heard of this as a pesticide. Benzene, which is an industrial solvent. So there are natural and synthetic molecules. Here's the issue. There are so many molecules and so little time. We don't have enough time to test all these molecules for cancer, for DNA damage. And you'll see short-term tests that have been developed in the next lecture. How do we proceed with public health? How do we regulate how much to expose people to if we're not sure if something is probably a carcinogen or possibly? So that's what I want to say about chemicals. Now, I want to next talk about radiation, which is potent in terms of causing cancer, and I'll talk first about ultraviolet light. The electromagnetic spectrum is the radiation that reaches the Earth. You've seen the visible spectrum before in the rainbow, but you may not realize is this has different wavelengths of light. Here are the wavelengths of light in the visible spectrum. Red has a longer wavelength of light, blue has a shorter wavelength of light, and the violet, the shortest, and the ultraviolet, which we really can't see, but you're going to see we react to in chemical terms, is very short. It's high energy. And when ultraviolet light hits DNA, something happens. What happens is if there are two T's, thymines, adjacent to one another in the DNA, just by chance, A, G, C, T, T, a bond forms between them, and the T's kind of bend over. This is a disastrous situation. Because what happens is, again, the DNA replicating machinery can't recognize this. So the machinery comes and says, well, I will put a T opposite the A. I'm replicating the top strand. A opposite the T, a T opposite the A, etc., etc. It comes to this thymine dimer, this thing that's got the 2 bases here. Here's the thymine dimer that's happening because of ultraviolet light. It's kind of bent over. The DNA replicating machinery says, wow, and puts in anything it wants just to keep going. So here it's going to put in a G and a C, and it just keeps going. And of course, you can see we're going to end up with a mutation here where we had 2 AT pairs, and now we have a GC and a CG pair. It's random which bases are going to be inserted. Again, a disaster in terms of DNA because the mutation becomes permanent. A very dramatic example of this happens when this type of mutation can't be repaired. If an individual has inherited a gene that cannot repair this type of system, ultraviolet radiation becomes extremely important. Now, ultraviolet radiation hits the skin primarily. You don't get it deep inside the body. It hits the skin primarily. And the typical skin cancer is relatively harmless, less harmful than most cancers. Typical skin cancer is squamous and basal cell carcinoma. As I mentioned, we don't even count them in cancer statistics. It's cured most of the times. But in a natural experiment, one that we wouldn't like to undergo ourselves, some people lack the ability to get rid of these thymines that are bonded to each other. And if these people lack the ability to repair, they are prone to skin cancer at a very early age. In fact, as teenagers, they will get skin cancer, whereas skin cancer comes usually much later in life. And so these people literally have to avoid sunlight. The second type of radiation I'll talk about is ionizing radiation. That's the radioactive stuff we talked about with Marie Curie and radium in the last lecture. Huge amounts of energy are released by radioactive elements in the laboratory. They're measured typically by a unit called the gray. The actual measurement's not that important. The old measurement was a rad. Typically, we are exposed to about 0.16 or a sixth of a gray in our entire lifetime of radioactivity. I want to just introduce therapy to you, radiation therapy, in this way. 60 gray over a period of a month. Now, we are normally exposed lifetime to 1/6. So we are being exposed to hundreds of times of the lifetime exposure radioactivity in a short period of time to repair a tumor. What type of damage does radiation do to DNA? It's a disaster. Here is some of the damage that radiation does. This is our radioactive atom. It releases both energy and particles, and both of them can cause damage to DNA. And I list some of the things. Hydrogen bonds break, dimers form like thymines. DNA can get cross-linked. The DNA molecules are together. They can't unwind. And they put in proteins that will cross-link. You get single-strand breaks of DNA, loss of bases, base changes, cross-links, and even double-strand breaks. The DNA, which is this huge, long molecule, will break. Right? And the DNA breaks very often are shown as chromosome changes. Because you remember at the very first lecture when I described the basketball player Kareem Abdul-Jabbar, and I described that in his leukemia cells there was a chromosome shuffling. Remember, a chromosome broke and swapped for another chromosome. Radiation damage will do that. It's so severe the DNA will break, and we end up with new chromosomes. And there are really two types of damages here in terms of chromosomes and cancer. In one is the one that we talked about in the first lecture. That is the formation of a fusion gene. That is half of one gene and half of the other because we shuffled genes and the chromosomes broke and resealed. The other way is a little bit different, and that is the gene itself, which is normally a quiet gene like a professor in the laboratory or whatever, all of a sudden is in a new environment. You know, put a professor in a nightclub, and they get very active. I'm not saying that normally happens. And they get very active all of a sudden. The gene gets turned on. So it's the translocation of a gene to an active site in the genome that causes it to be turned on. We will see examples of this later on in the course. So these are the carcinogens that damage DNA. I mentioned there are some that don't damage DNA, and I'll describe three of them. First, estrogen. You're familiar with this. Second, asbestos. And third, bacterial infection called Helicobacter pylori. So let's first talk a little bit about estrogen. You know from all the epidemiology we talked about earlier on in the course, estrogen causes cancer. How does it do it? A signal, which estrogen is, enters the cell, binds to a receptor, which you're familiar with already. We talked about that previously. The receptor is the door, this thing on the door that binds. It's not in the door of the cell, but it's inside the cell. It binds to the estrogen, and the combined couple, that is the estrogen and its receptor, are escorted into the nucleus, where they stimulate indirectly the cell to divide. And this happens every month in the uterus lining of the ovarian cycle. Cancers that arise, endometrial cancer, we know that already. Breast cancer, we've seen evidence for that. Ovarian cancer. This especially happens if a cell overexpresses through some genetic mutation, again, the receptor for estrogen. And of course, treatment is going to involve manipulating the system, the estrogen receptor system, to try to get to reduce estrogen binding and stimulating cell division. The second non-DNA damaging substance I want to talk about are minerals. Minerals cause cancer by stimulating cell division. Asbestos is a widely used mineral. It's used in buildings, pipe, the paper industry, ships, brakes of a car, etc. People inhale the mineral, gets down in their lungs, and when it lands on the lining of the lungs, causes a specific cancer, which is rather interesting. It doesn't cause any other type of cancer. This cancer is called a mesothelioma. So it's called a signal cancer for this type of damage by asbestos. It doesn't happen with other carcinogens. It's specific to asbestos. I indicated in the last lecture, In shipyards, people exposed to asbestos would get cancer exposure after 20 years or so working in a shipyard. So they're insoluble things that cause cancer. Now, in reality, what's going on? They're damaging the tissue, and in themselves, they're not damaging the DNA. But they're causing the tissue to try to repair itself and divide and divide and divide to repair the damaged tissue. And when a cell is rapidly dividing, it will accumulate spontaneous mutations or accumulate mutations from other carcinogens. Randomly, that happens. A third thing I want to talk about that's not DNA damaging directly is an infection. And the infection is in the stomach, and the bacterium is called Helicobacter pylori. Helicobacter pylori causes ulcers. It's a very famous Nobel Prize-winning work by Barry Marshall in Australia. People used to think ulcers were caused by stress, bad eating, etc. It turns out, no, they're caused by an infection, a simple infection with a bacterium. And Dr. Marshall was quite famous in the sense that people really didn't believe this. They had this long tradition of saying, no, 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 it's diet. And he said, well, I've cured people using antibiotics. Get rid of the bacteria, and you get rid of the ulcer. And so he swallowed some bacteria and started getting the ulcers. And then he, of course, treated himself to make sure he got better and didn't get cancer or didn't get ulcers. I want to go over the evidence for this because it really recapitulates some of the work we've described in epidemiology. So this is evidence for a bacterial infection causing cancer. Case control studies have been done. The relative risk typically is between 2 and 8. That is, you take a group with stomach cancer and a group without, and you see which ones are infected with Helicobacter, and there are more. Cohort studies have been done. You take a group of people with cancer and without, and you ask which ones are infected. And typically, they took a group of people, they did it in a longitudinal way, who were infected with H. pylori for 8 years, and 6% of them developed stomach cancer, and none of the uninfected people got it. That's an example. If you want to prove something causes cancer, you have to do a laboratory experiment. Here's the laboratory experiment. They took uninfected laboratory animals, infected it, and they got stomach cancer within 18 months. Fourth, let's get rid of the thing. If you get rid of the thing, you'll have less cancer, right? From epidemiology. Remember the Broad Street pump? Got rid of cholera. Here we're getting rid of cancer by wiping it out with antibiotics. And if you follow them, there's a severe reduction in stomach cancer. So this is an amazing system. The H. pylori bacteria, and here's a photograph of them, infect the stomach. They live in the acidic environment of the stomach. And we now have a very precise molecular description of the events involving the bacterium, sending signals out that stimulate the cells just like estrogen does, only now it's the lining of the stomach. Next, I want to turn to epigenetic changes in DNA. Epigenetics is possibly inherited, but it's changes in DNA that not necessarily inherited that can contribute to cancer by changing the expression of DNA. Remember, DNA sits inside of a cell, and it's going to do nothing unless it's expressed in some way. So I have many, many genes in my body. For example, in my hair follicle, it's expressing a bunch of genes. The genes that are being expressed of the 24,000 in the genome, a couple of thousand just to keep the cell going, and then the gene for making hair. Remember, the hair protein keratin is expressed in the hair follicle cells. But the gene for making liver or the gene for making hemoglobin is not. You don't see hemoglobin pigment in my hair. The gene for making fingernails is not. You don't see fingernails growing out of the follicles either, right? So there's selective gene expression. Now it turns out that this gene expression can be regulated in a highly specific way, and we know what that is. This is called epigenomics. How do we know that this gene expression can differ? Consider identical twins. They are identical genetically. At age 3, if you look at which genes are being expressed in their brain, it's identical. The same genes are being expressed. By age 50, different genes are being expressed, all largely due to epigenetics. Not because they learn something differently, but because of epigenetics. Some of these genes are ones that are very important for the structure of the brain, they don't get changed in learning. And these genes will be expressed differently because of epigenetics. What are the epigenetic changes in cancer? There are 2 of them, and I will discuss both of them. These are semi-permanent changes in DNA for its expression only. They do not change the basis of DNA at all. They're just adding something a little bit that's not changing it. It's not like a molecule of aflatoxin coating the DNA, or nitrous acid changing the DNA, changing NH2 to O or something like this. No, it's just a small change. The first is called DNA methylation. DNA methylation turns genes off. It's a signal that says, pile on, turn me off. Don't let me get expressed. And there are two types of DNA methylation. If you reduce the expression of a gene that normally stimulates cell division, tamp that down, then the cell's not going to divide, less cancer. If you reduce the expression of a gene that normally blocks cell division, so now you're tamping down the inhibitor, cells will be allowed to divide. The other method of epigenetics is called DNA packaging. And we'll talk about that briefly in a moment. And again, we're going to look at these two types of genes. If we increase the expression of genes, so now we're opening up DNA, we're stopping it from being blocked, increasing the expression of genes that stimulate division, of course, you're going to get more cancer, right? It's going to be allowed to go ahead. If we stimulate genes that block cell division, of course, you'll get less cancer. So you can see that there are reciprocal things going on with both of these mechanisms. Let's look at methylation. Methylation happens with a group called a methyl group. You don't need to be a chemist, but I'll show it to you anyway. It's this carbon with 3 hydrogens, and it's put on one of the 4 bases of DNA C. And when that group is on there, all sorts of molecules go in and say, shut up. Don't say anything. Do not transcribe. Do not get expressed. Don't make anything. Okay? In cancer, of course, several things are going to happen, right? If a gene is methylated, you could take, as I mentioned, a gene that inhibits DNA normally, like a tumor suppressor, and you could silence it. And if you silence the brakes, then cells will divide. On the other hand, you could turn off oncogenes, right, by methylation as well. And again, you're going to inhibit DNA. Is this relevant clinically? Absolutely. Absolutely. We can use DNA methylation at the time of diagnosis of a tumor to determine whether aggressive treatment is needed. So here are some examples. These are tumors. There's a list of tumors with increased DNA methylation of genes that normally, and it blocks genes that inhibit cell division. So these are normally tumor suppressor genes. They normally block cell division. We methylate them, so we block the blocker, and now cells can divide. And there's a whole list of cancers here where this has been found. And if you find something like that, it means it's correlated with a highly aggressive tumor, and so you're going to want to do highly aggressive therapy. The second way of doing epigenetics is DNA packing. You remember this diagram from earlier on? Maybe you do, maybe you don't. You have DNA. Remember, 6 feet, 2 meters of DNA in a cell, getting scrunched down terrifically. And now if you want to make DNA available to get recognized and expressed in some way, you want to open it up a little bit, let's undo it. Let's take that mechanism and undo it. Loosen the packing, right? I'll let DNA be exposed to the expression machinery. So DNA is usually packed tightly by proteins, and if you change those proteins, you can change the expression of DNA. This is in the clinic now. There is a drug called, which is called vorinostat that has been developed that targets the DNA packaging mechanism. And it does it by causing the DNA packaging to loosen up so that genes will be expressed. And what genes do we want to express? The genes that inhibit cell division. So you treat a patient with this drug, the genes that stop cells from dividing will be expressed in greater amounts and will tamp down the tumor. This is, you can see in a CT scan, CT scans of a lung cancer, for example, will show a significant reduction in tumor if they are treated with these drugs. These are called HDACs, histone deacetylase inhibitors. There are many of them now under development and under clinical testing for cancer. And the idea, again, is to open up the DNA of inhibitory genes, genes that normally block or put on the brakes for cell division. So in this lecture, and the lecture previously, I've discussed DNA and DNA damage. First, in the previous lecture, I introduced DNA, and in this lecture, I talked specifically about carcinogens that damage DNA. The carcinogens I talked about were chemicals and radiation. I gave you a couple of specific examples, and of the damage that they do to cause cancer. Then I discussed a couple of carcinogens that don't damage DNA. And finally, I discussed these epigenetic little changes in the expression of DNA and how that can lead to cancer, and they're being targeted by drugs. Now, epidemiology identified carcinogens. The past couple of lectures I said how they work on DNA or whatever to get cancer initiated. But here's a problem I brought up earlier in this lecture. We are exposed to thousands of chemicals and substances every day, and we can't test them all by long-term epidemiology or studies in carcinogens. So now I'm going to set that problem up and say, what do we do about this in terms of public policy? In the next lecture, I'll talk about how we can deal with this issue of overwhelming cancer-causing things in the environment. Thank you.